Good afternoon, everyone. Today's presentation focuses on postmortem staining. It's often confused with liver mortis, but chemically and practically is very different. Um, Instead of just being pooling blood, it involves the hemoglobin actually leaving the vascular system and reacting with the tissue proteins and becoming permanently fixed. That's why it's called extravascular lividity. Um, the chemistry, understanding it is very important and critical for embalmers because once the staining gets in, regular arterial injection is just not enough to correct it. So when we're talking about staining, we're referring to it as like a true extravascular discoloration. Early lividity, liver mortis, it's still inside the vessels and can shift with repositioning. The staining, it cannot. That's why it's a stain. Once the hemoglobin breaks free from ruptured red cells and it bonds with the tissue proteins, the color becomes part of the tissue itself. If you can reposition and the color moves, it's liver mortis, and if it doesn't, it's actually chemically locked in. The development of the staining, it's three main stages. First, it's early liver mortis. Gravity pulls the blood into areas of the body. And then it's just the passive settling is what happens first early on, liver mortis. Second, the cells begin to break down, membranes lose integrity, and the hemoglobin inside them begins to spill out. And third, the free hemoglobin now diffuses out of the vessels and starts reacting with nearby proteins. By this point, it's no longer just a pool blood, it's actual altered tissue chemistry. No matter how you arterially inject or type to drain or try to drain, it's never going to reverse it because it's chemical change. Environmental and physiologic factors. Temperature is the biggest influence. As we all know, cooler environments slow down the cellular breakdown. Warmer ones speed it up, pushes hemolysis and the pigment formation faster. Time of death matters. Staining becomes permanent between 8 to 12 hours usually. Also, this can vary depending on the environment. Rigor adds a big layer because the contracted muscles can compress vessels and it traps the blood pooling in place and giving it more time to break down and diffuse. Finally, changes in pH and dehydration can shift the tissue chemistry as well. This just increases the reactivity with the pigments and in some cases can make the staining worse. So interaction with other postmortem changes. As the decomposition advances, the chemistry gets obviously more complicated. The hemoglobin breakdown, it forms pigments like hematin, which lowers the tissue pH and makes formaldehyde less effective. This is really important for anybody embalming. When hydrogen sulfide is present, you can also see the greenish tones that you can sometimes see. This is, bear with my pronunciation here, sulfahemoglobin forming because these chemical reactions overlap. And what can start as like a red-purple stain can shift colors and, intensify depending on how far along in decomposition you are. Embalming challenges. So the biggest challenge with staining is that the pigment is no longer inside the vascular system. Arterial fluid cannot reach it, can't push it out, you cannot drain it. It's in the tissue itself. Not to mention those stained areas are now acidic and often react with the formaldehyde by producing embalmer's gray. So even if your injection, you have the proper distribution, proper pressure, you're just fighting the chemistry of the tissue at that point. And that's why it forces embalmers to have to think about alternative techniques. All right, strategies. So once the staining is present, adjusting the chemistry of the fluid that you're going to inject becomes essential. We got to use higher index fluids to overcome the acidic tissue conditions. Buffers can be used to help neutralize that environment. Pre-injection chemicals, we can use that for clearing out the vascular system before the main injection, and co-injection agents help improve penetration into these tissues. None of these are magic fixes by any means. They just improve your results. You have mechanical assistance, massaging, intermittent drainage, repositioning, all things that can improve the distribution when the staining, like before it becomes fully fixed. In cases though, let's like where the pigments are chemically bound, the only realistic improvement comes through local treatments, bleaching agents, phenol packs, controlled cauterizing compounds. You can use these to lighten, camouflage the discoloration, try and blend it more. These are all super important things to take into consideration before the viewing, during the viewing. Oftentimes, it usually comes down to cosmetics. All right, case study time. So we got a body here. It was discovered about 18 hours in a cool environment. By the time they go to embalm, all of the staining, it's not blanching at all. So it's fully fixed, or almost fully fixed. Fluid distribution, it's normal everywhere except for in the stained regions. Of course, this is going to give you embalmer's gray. This is because the formaldehyde is reacting with the stained tissues. Phenol packs lighten the color. The correction was incomplete because the pigments were chemically bound. So again, you really can't do a whole lot. Just kind of comes down to cosmetics. Practical checklist, key takeaways. So, postmortem staining, definitely not the same as lividity. It's a chemical change, not a positional one, or physical one, whichever. You have to catch the body early to prevent it. That's not always a luxury. And once the staining is fixed, best results come from combining adjusted arterial chemistry, local treatments, and just kind of being realistic with the families about what can't be corrected. Understanding the underlying chemistry isn't just academic. It directly guides the decisions we make. And it's really necessary for make sure we deliver the most presentable results. So, yeah.